Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT class 12th molecular basis of inheritance, that is part 7. The machinery and the enzymes. Let's see. Here all living cells such as an Escherichia coli, the process the replication is required a set of the catalyst enzymes. So the process of the Escherichia coli, the replication process we require as the number of enzymes. Am I right? The main enzyme is referred to as a DNA dependent polymerase enzyme. That is what DNA dependent DNA polymerase enzyme is very important for the process of in the replication process. Am I right? Since it uses a DNA template to catalyze the polymerization of in a their deoxynucleotides. So within the DNA template, they having the number of their the deoxynucleotides is present. They get participate as in that in a replication process. Next, this enzyme are highly efficient enzyme. They get have in a catalyst polymerization the large number of the nucleotides has in a very short time. So it is in a long DNA. Already you know the DNA is in a very long, right? In this DNA they having as a number of here nucleotides as present. So in these nucleotides immediately they get completely to get synthesized. Now, for example, they given here Escherichia coli that has only 4.6 into 10 to the power of there are 6 base pairs that compared with is in a human host their deployed content there 6.6 into 10 to the power of 9 base pairs. In completes the process of the replication within 18 minutes. So they're completely that Escherichia coli they complete the life they complete the replication process within 18 minutes. Very important is they complete as in 18 minutes. Let us hear. That means their average rate of in that polymerization has in that approximately that 2000 base pair per second. So around as in we compare here how many base pairs the 2000 that base pairs or to completely they get they participate in that in polymerization of the DNA. Next, here not only do this polymerase having very fast, but they also their catalyst their reaction is a high degree and accuracy. So in this total process, how that takes place here? The number of enzymes they get participate, right? Here the number of enzymes they get participate within a accuracy means they're properly they're working. Then only we are getting as a replication process, right? Let us any mistake during the replication would be result into as what mutation. So if in case here they get guanine is present, here they having a cytosine actually, right? If in case in that place they get as an adenine, what happened? That takes place as a mutation. Okay, now so the total the replication is takes place as in a very accuracy. Any small mistake is takes place, what happened? It is complete the collapse means that takes place as a mutation. Okay, now the furthermore the energetically the replication is very the expensive process. The deoxyribonucleotides, the triphosphates, they serve as a dual purpose. So here most of the triphosphate and deoxyribonucleic acids, these are completely they participate as in what in a they participate as in a dual role. They participate as what in a dual role. And in addition to acting as a substrate, they are in provide energy for as a polymerization reaction. That is in what is the polymerization reaction? They get participate in two ways. Two ways. Yes, that is the two terminal the phosphates, the deoxynucleotides and triphosphates in a their high energy the phosphates the same in the case of in a ATP. So the simply when they get this total process, how do they takes place? When they get using the utilization of any some energy and then complete they get participate in that in a formation of the incompletely the replication process. Okay, next for example, next in addition to the DNA dependent DNA polymerase is mainly additional enzyme or is required in complete process of replication within a high degree of accuracy. So mostly just only we say what in this in a total DNA is there. This total DNA is very long. So in this total DNA, the replication, the process, it is going on. It is very long stand now. So the total replication is takes place in a very accuracy and they properly they going. How if in case they are completed enzyme they are participated properly, then only this work will be there going on in one process. Now for long DNA molecules, since there are two strands, the DNA cannot be separated in entire the length of due to as in a very high energy requirement. So the replication will be going on here. So this is a long DNA strand. So in this long DNA strand, the replication will be get a continuous process. What happens? They requires more energy. Am I right? And the replication then occurs within a small opening that is in a DNA helix, refers to as a replication fork. So in this total DNA, the total replication is not takes place. Only a segment of the region that they get start with as a replication. Here has it is a this process. For example, is in a DNA. Am I right? In this DNA, only a segment there is this region they can start with as a replication. Okay, like now this part is called as what? This is completely called as a replication fork. It is what? It is in a replication fork. Is what? Replication fork. Now here the DNA dependent DNA polymerase catalyst the polymerization only one direction, that is in a 5 prime to as in a 3 prime direction. So see, mostly when you observe here the replication process, when you see here the mostly the replication process here, what happened, the direction will be the going as an either 5 prime to as an 3 prime, 5 prime to as an 3 prime. Here this is the 3 prime to as an 5 prime. Am I right? So these are completely called as what? This is completely called as a template strand. Am I right? Now here this creates some additional component that complications it has in the replication fork, that consequently one of them is start from the template, the polarity is 3 prime to 5 prime. So one polarity, so the template polarity they comes as what? They comes as an the 3 prime to as an 5 prime. The it is in the replication is a continuous, while the other the templates they get 5 prime to 3 prime, that is in a discontinuous. So it is the most, for example this is there, it is in a completely in a the discontinuous stand, it is in a continuous stand. One is in a discontinuous stand and another one is in what? In a continuous stand. So for example in this discontinuous stand, how do they get again bind? With the help of the enzyme is, that is in a DNA ligase enzyme, that is in what? The DNA ligase enzyme. See here the discontinuously the synthesized fragments also later joined by the enzyme is DNA ligase. If the DNA ligase to get help to get joined with as in all fragments, they finally get as in that another stand. Okay, that is. Next, here the DNA polymerase is their own, cannot be there initiate the process of the any replication. Also the replication does not there initiate the randomly at any place the DNA. So it is not randomly takes place. It is one side that completely takes place as in a, say for example it is not randomly takes place. Only for example first here to here the first the replication will be done. After here to here the replication will be done. After is complete, after they can start here to here the replication will be done. Like only this is parts on the like this one, this like the replication will be there takes place. There is the definite the region of Escherichia coli DNA where is the replication there originates. So where they get replication will be get start, they get start the replication. Such regions were terminally called as an origin of replication. Where they get the replication will get start. So these regions are called as what? It is an origin of replications. Origin of, that is in a replication. Origin of replication regions. Okay, like next, it is because of this in a replication. See the because the requirement of the region of the replication, that is in the piece of the DNA is need to be as in the propagated during the recombinant DNA process. They produced as a requirement as a vector. The vector provide as a region of the replication. So in one particular site, in that one vector here, any one vector they having as in a particular the specific location, that is in a ORI site, that is in a ORI site, they completely get start with as in that number of the strands will be get produced. The number of strands will be to get produced. So here they given as an a the 5 prime to as an a the 3 prime and 3 prime to as an a 5 prime. They given as the one stand. See mostly their 5 prime to as an a 3 prime they given as in their continuous stand, that is in a their leading stand, that is in a leading stand, and the 5 prime to as in 3 prime. Before I said that is in a discontinuous strand, that is called as a what? Lagging strand. That is called as what? Lagging strand. Okay, here the completed DNA ligase enzyme to get participate with the help of as an A to get attached. Okay, next. After further now the every detail the replication is understood well. In eukaryotes, the replication, the DNA takes place in a S phase of the cell cycle. Very, very important. Mostly here, the DNA replication takes place in where? In the DNA, yes, in eukaryotes, S phase, that is synthesis phase. In eukaryotes, in that in eukaryotes, in the DNA takes place, synthesis takes place in which phase? That is in a S phase, or nothing but as in a, that is synthesis phase. Okay, next. The replication of the DNA and the cell division should be there highly, they are coordinated. A cell division and the replication is very coordinately that takes place. A failure of the cell division after the DNA replication result to as a polyploidy chromosomal anomaly, means that total that takes place look like any mutations. Okay, and you will learn detailed the nature of any origin and the process of any occurring at this site in a higher classes. This total information discuss in that in a higher classes. Okay, this is in total information about as the DNA replication. Okay, the remaining to discuss in next class. Thank you for watching. Please share, subscribe and like.